Peptic ulcer. What does ulcer means? Ulcer means discontinuation or break in the lining of the mucous membrane or skin. Now, peptic ulcer. Peptic ulcer occurs in the part of gastrointestinal tract which is exposed to HCl or pepsin, means stomach or duodenum. Now, what is the pathophysiology? The pathophysiology can be due to imbalance between aggressive and defensive factor. Aggressive means which causes harm to the stomach mucosa, and defensive which protects it from the aggressive factors. What are aggressive factors? Acid, pepsin, bile, H. pylori, they are included in aggressive factor, and defensive factor includes gastric mucosa, bicarbonate, prostaglandin, nitric oxide, high mucosal blood flow, innate resistance to mucosal cells. Secondly, it could be infection of Helicobacter pylori, which could cause the peptic ulcer disease. Now, peptic ulcer can be gastric ulcer or it can be duodenal ulcer. Gastric ulcer is majorly due to deficient mucosal defense with normal or low acid secretion. In duodenal ulcer is mainly due to high acid secretion. Now, to treat the peptic ulcer, the treatment should include reduction in acid secretion or neutralizing the acid or removing the cause. Example as Helicobacter pylori. Peptic ulcer could also be caused by NSAID, stress, smoking, etc. Now let's learn about regulation of gastric acid secretion. Gastric acid secretion is mainly the final step includes H plus K plus ATPase located in the parietal cells. There is a terminal enzyme known as H plus K plus ATPase, which is also known as proton pump, which secretes H plus ion in the apical canalicule of parietal cell and in exchange of potassium ion. And the potassium and chloride are released with the K plus Cl minus symport. So Cl minus is also secreted and H plus is also secreted, which leads to secretion of HCl. Now the H plus K plus ATPase requires ATP for its action. It is stimulated or activated by mainly three receptors, which includes muscarinic M3 receptors, histaminic H2 receptor, and CCK2B or gastrin receptors. These two mainly activate the proton pump. The muscarinic receptor mainly act through IP3, IP3 DAG pathway, and histamine has the dominant role in activating proton pump. CCK2B acts via IP3 DAG pathway. Now let's see how these three receptors are activated. Firstly, there is a G cell locator which secrete gastrin in the bloodstream, and from bloodstream gastrin reaches the parietal cells. It is not directly secreted. The gastrin acts by directly stimulating CCK2B receptors. It can also stimulate CCK2 receptor located on histaminocytes or enterochromaffin-like cells or located in oxantic gland, and can increase the release of histamine and indirectly cause the activation of proton pump. Now, the ECL or enterochromaffin-like cells also contains muscarinic receptors. They mainly release histamine. They contain histamine granules and release histamine, which activates H2 receptor. Now, whenever we eat food, it gives signal to the brain, and through brain the signal is carried by vagus and acetylcholine is released. This acetylcholine stimulates uh, the uh, enteric nervous system ganglion cell, which has nicotinic and muscarinic M1 type of receptor. Remember that in ENS ganglion cells there is located M1 subtype of receptor, and in parietal cell there is M3. Majorly it acts through M1. Now it stimulates this. It causes release of gastrin-releasing peptide and acetylcholine. Acetylcholine can directly act on M3 receptor, or it can bind to the muscarinic receptor on histaminocytes. Gastrin-releasing peptide activates G cell and causes directly release of gastrin. Now we saw the activation of these three receptors. Now in defense, there are D cell located, which releases somatostatin, which is a hormone which inhibits gastrin and histamine release and causes decrease in HCl secretion. Helicobacter pylori infection causes destruction or damage to somatostatin, it causes damage to the D cell, which causes decrease in somatostatin, which indirectly causes increase in HCl. Now there are mucus cells present. Mucus cell secretes mucus and HCO3 minus bicarbonate ion, which protects the mucosa or mucosal lining of GIT from the hydrochloric acid. It contains prostaglandin receptor, the arachidonic acid, which is acted upon by COX enzyme, and it is converted into prostaglandin E2 and I2, which inhibits HCl by opposing cyclic AMP generation and inhibit gastrin. Now here I forgot to tell you that M3 and CCK2 act by IP3-DAG pathway and histamine acts by cyclic AMP pathway. Now the prostaglandin is mainly has a cytoprotective role in gastric mucosa. As we saw over here that it increases the mucus and bicarbonate secretion. This secretion added to the mucosa and prevent it from damage by the HCl. The secreted HCO3, it neutralizes the H plus ion, so it acts as a barrier for HCl. Now, what are the goals of anti-ulcer therapy? The primary role is to relieve the pain, ulcer healing, prevention of any complication like perforation or bleeding, prevention of its relapse.